Hey, this is Mrs. Dellosso, and in this podcast, we're going to talk about mutations, and specifically, we're going to be talking about changes within the genes. So let's refresh our memory. Gene mutations are a change in the sequence of bases in a gene. So here, if we take a look at our DNA molecule here, looking at all these bases here, this could represent a single gene. So when we're talking about genetic changes, we're talking about changes that can occur in any of these bases here, whether if we switch a base or if we add another base in here or delete one, and we're going to see what effect it can have on the chain of amino acids or the proteins that get produced. So point mutations. When only one or a few nucleotides are changed, it is called a point mutation. Even though a change in one nucleotide may be an insignificant change, the effect on the organism can be profound, because even if one nucleotide is changed, that could ultimately change an amino acid, which can change the whole protein, either making a protein not function properly or maybe not even making the protein at all. So point mutations can occur in one of three ways, either a substitution, addition, or deletion of nucleotides. And we're going to take a look at examples of each of these. So let's just take a look at an example of this just using words and sentences which would represent the genes and the proteins. So let's take a look at one base change here. Again, meaning it could potentially change the whole meaning of the protein. So let's say we have this gene here, and let's break it out into its three bases or the codons. Very similar to what we did with the messenger RNA. So if you read this, it says the fat cat and the red rat ran. That's the sentence, right? But let's say we have a change in the sentence or in one of the letters. Let's say the T is changed to an R. Now let's see the effect that it'll have on the sentence. Or let's even change a different letter and let's see. So when you look at these two sentences, just changing one of the letters changes the sentence completely. So that's the same thing when we start looking at DNA. Even if we change one base, it could potentially change the whole protein. So let's take a look at a substitution. Substitution occurs when a nucleotide in the DNA sequence is removed and then replaced with a different nucleotide. So let's take a look at this DNA sequence here. So if you take a look, these first three DNA bases, right, if we do transcription, we would have the UGG forming the messenger RNA, and then that would code for the tryptophan amino acid. Then the next codon would code for this amino acid, followed by this one here. Now, let's say we do a substitution change. Let's see what that effect would have. So let's say we swap out that C nucleotide with a G. Now take a look at the amino acid that gets formed. And if we go back to the original, notice how it changed. So even if we substitute one nucleotide, that's going to change or could change the amino acid, which would then might have an impact on the protein that gets made. Sickle cell anemia is one of those disorders that's caused by a substitution. So sickle cell anemia is caused by substituting adenine for thymine in the DNA molecule, resulting in a defective form of hemoglobin, and hemoglobin is the protein that carries oxygen. So here, if we were taking a look at the normal red blood cells here, the structure of the normal hemoglobin and the amino acid sequence here, and if we take a look at the sickle cell, here we can see that different amino acid that pops up, and that's because there's a change in the bases that codes for the protein or the specific amino acid. So notice here at 6 it's supposed to be glucine, but with the mutation or change in the DNA, we now produce valine, which would give the cells this shape here. Now, why is that bad? Well, because a person with sickle cell, the shape of the blood cells are changed, which makes it harder to flow throughout the body, which can give a lot of people a lot of pain. And there's also problems with carrying oxygen throughout the body as well. Up top here are the red blood cells, the normal shape, and they're able to flow a lot more freely than if they were the sickle cell shape here. And you can see they can get stuck here. All right, let's take a look at nucleotide additions and deletions. So a nucleotide addition occurs when an extra nucleotide is placed in the DNA sequence. So since the codons contain 3 nucleotides and they're read that way, adding a nucleotide to the DNA shifts all the nucleotides down by 1. This is called a frame shift. And the same thing is going to happen if we remove the DNA. So a nucleotide deletion is the opposite of addition. So a nucleotide is removed from the DNA, and that can also result in a frame shift. And we're going to take a look at a frame shift right now. All right, so frame shift by adding or deleting one or more bases, and that could also change the meaning of the protein as well. So let's take a look using our words here as an example. So here we have the fat cat and the red rat ran, right? Now let's see when we add a letter in here how that's going to change, if it does change the sentence. All right, let's break it out into those 3 letters again. So does this change the sentence at all by adding a nucleotide? It sure does. So what about if we were to delete one now? Delete one of the letters. Let's take a look at the sentence. So does this change the sentence when we remove a base? It sure does. So that's a frame shift. What happens when we add a base or delete a base? It causes the sentence to be read differently because we have to now shift the bases down. Because remember, when we formed proteins, they're read in bases of 3. Those 3 messenger RNAs. So if we're squeezing one in there, it's going to cause an effect on the rest of the nucleotides that will be read. Alright, so let's take a look when we're actually using bases here. So addition, let's add a base. So here's our sequence here. Alright, so here's our messenger RNA at the top. These are the following amino acids that it codes for. Now we'll add the base here. Now we're going to add U in between the G and the C. Let's see what happens. Take a look. So, if we were to compare these amino acids with the top one, you can see a big change in there just by adding a single nucleotide or a single base here. So it's definitely going to change the protein, and that's a frame shift. Let's say when we delete. So here's our messenger RNA followed by the amino acid sequence or chain, which would be the protein. Now we're going to delete the base between the G and the A, so that we're going to delete that C. Let's see what happens. Alright, let's take a look at the amino acids and compare it to the first. So notice, again, does this protein change? Yes, it does. Again, because we're comparing the 2 chains of amino acids. Again, there's a major change when we add or delete bases. Alright, another disorder caused by a change in a gene is cystic fibrosis. So cystic fibrosis is a genetic disorder that affects the respiratory and digestive systems. People with cystic fibrosis inherit a defective gene on chromosome 7. So here's that defective gene there. It's the CFTR gene, which stands for cystic fibrosis transmembrane conductance regulator. So the protein produced by this gene normally helps salt, which is sodium chloride, move in and out of cells. So over here we have a normal CFTR channel, so salt can easily move through this channel here. If the protein doesn't work correctly, then the movement is blocked, and normally thick, sticky mucus is produced on the outside of the cell. So here is a defective or mutant CFTR channel, and because it doesn't allow the salt to easily pass through, that thick mucus builds up there, and that's a problem because this mucus clogs the airways in the lungs, and it increases the risk of infection or bacterial infections there. Not only does this happen in the lungs, but this can also have an effect on the digestive system. Cystic fibrosis can cause problems with how people absorb nutrients. And it can also affect the sweat glands as well. So this is a recessive disorder. So that means that both parents need to carry this gene and they'll pass it on to their offspring, and that's how they will inherit it. And again, this is caused by a mutant gene, a change in nucleotide basis. Alright, so that sums up this video on gene mutations. If you have any questions, please ask me.